Okay, I guess I can do a short introduction, and then we can start the presentation. So it is my a pleasure and my honor to introduce to you Professor Carlos Brody. Carlos, he is born and raised in Mexico, and has a PhD in neuroscience from Caltech, and has been basically mentored by Ranulfo Romo as a postdoc, and also by Tony Mofshan. He initially was in Cold Spring Harbor, but now he's in Princeton for I guess more than 10 years or 15 years. And he's had basically collaborations with Zach Mainen and Tony Zador, and basically he has a very good knack of training non-primates to do complicated decision-making tasks. Very strong on both computational side and training. He has become an investigator of Howard Hughes Medical Institute from 2008, and he uses a combination of high-throughput behavioral training, neural recording, and perturbation methods to investigate cognition, decision-making. I have to say he's one of the, I would say, brightest neuroscientists I encountered when I was in the U.S., and I look forward to a very, very high-quality and educational talk from him. Very happy to have him in the symposium. So, Carlos, we cannot see your presentation now. Would you be able to share one more time?
Yes, here we go.
Oh, thank you so much.
Thank you, and thank you for that tremendously kind introduction. I'm not sure that I can live up to that, but well, I'll give it a shot and let's see how it goes. I'm looking forward to your questions. I think I won't be able to see your questions until the end of the talk, just because of how I'm using Keynote, and Keynote takes over the entire screen. If there's a burning question, please, if there's a burning urgent question, please help me by saying so aloud. Okay, so my lab, broadly speaking, is interested in what we call the neural architecture of cognition. And when I say that, I have to explain kind of like, what do I mean by cognition exactly? And what I mean is processes such as these. Working memory. How do you keep something in mind for a few seconds and manipulate and use that mental representation? Planning. How do you use knowledge of relationships between actions and outcomes to choose what to do? For example, if you know that moving a pawn to a particular square on the chessboard will put pressure on another piece, how does that affect your choice of which piece to move? What is sometimes called executive or cognitive control, which I like to describe as how you control how information is routed within your brains. For example, I could say to you, today, red means go and green means stop. And you could go out and you could drive like that. And it would take a little bit of effort. But somehow, just by deciding to follow those instructions, red, instead of being routed to the brake pedal, would now be routed to the accelerator pedal. How can you flexibly control that routing? And evidence accumulation for decision-making. How is it that you can accumulate evidence for or against different decisions and use that accumulated evidence to commit to a choice, to commit to a decision choice and implement it? These are the kinds of things that my lab is interested in and that we've been working on. And we're interested in them at the single cell resolution level. In other words, we want to know how networks of individual neurons interact with each other within and across brain regions in order to implement these kind of cognitive processes. And in order to do that, it's difficult. It's very rare to study that with humans. So you have to study it with animals. You have to do invasive neuroscience and in order to be able to record single neurons. And that means that you have to train animals to perform these tasks. And typically the process is that we try and develop some well-controlled behavioral task. Well-controlled in the sense that it allows us to isolate which processes are involved and when, so that we can study them carefully, both from the behavioral point of view, and once that's established, then from the neurophysiological point of view. And there's a very slow part with that, which is training these animals on the well-controlled tasks. We can't tell the animals what the task is, and so that means that we have to slowly train it along, and that can take a very, very long time. when my lab started, most of these kinds of questions at single neuron resolution were addressed using non-human primates. But as was mentioned in the introduction, I was at Cold Spring Harbor Labs at the time. I had a computational lab, and my neighbors were Tony Zader and Zach Mainen. And they had started trying to train rats and mice in tasks that were similar to the kinds of tasks that were up to then mostly developed for monkeys. And the idea was that the rodents would be so much more tractable if we could train them on these tasks that it would be much easier to investigate the workings of the brain using those animals. And I got totally fascinated by that, and I joined them on the effort. And since then, my lab has been both a combination of computational and experimental efforts. And as I said, the motivation for doing this is that rodents are, compared to non-human primates, compared to monkeys, they're relatively small. They're inexpensive. You can get good behavior out of them. A big question was to what extent the behavior would be sophisticated enough. And very importantly, they're amenable to modern molecular tools. Of course, nowadays there's many labs that are pursuing this, but back then this was kind of like the beginning of it. We were part of a wave of labs that were starting to do that. And here in this picture, I'm showing you a picture of the behavior room in my lab. And this is a computer rack, so a person who's about that tall. And so you're seeing 9 out of 36 behavior rigs that we have in the lab. And we train 9 rats per rig per day. We do 9 shifts per day. So we train about a little bit more than 300 rats every day, 300 individual animals every day. That gives us a lot of statistical power. That gives us about 1 million behavioral trials per week. And importantly, it allows us to parallelize the training. So for example, if there's a postdoc in the lab that has 40 out of these 300 slots for a particular task that they're studying, and so they're training 40 animals in parallel, and the task is one that requires 4 months of training, then on average, thanks to that parallelization, they get 1 trained animal out every 3 days. And that's a completely different world to training just 1 or 2 animals a year. Now, in order to train this large number of animals, you have to train them in parallel. You have to automate the code for enabling that parallelized training. There's no possible way that a person is going to sit in front of each of the 40 animals simultaneously and give them a little bit of water here or there to encourage them to do this. The whole thing has to be automated. That in turn has a lot of advantages. It takes a lot of effort to develop the automated code that will train the animals in the tasks that we want to do. The animals often do things that we don't expect, and so we have to find that out by trial and error. But once we have code that works, then parallelizing is just a matter of copying the code. So that's fantastic. And also, that really documents how the training happens. So that's also very useful, and it's something that can be shared. Now, as I said, we were part of a wave of labs starting this, and since then there's been many others trying to study cognitive questions in rodents. There's many labs doing it. Our particular interest and focus has been on what we sometimes call complex or higher cognitive processes. And I put the higher in quotes really to mean that I don't really know what higher cognition is. In practice, what I know is that there are tasks that it takes a lot of time to train the animals to do. There are sophisticated tasks that it takes a lot of time. And when it takes a lot of time, for example, 4 months per individual animal, this parallelized system is really, really helpful. It really makes a difference. So using that system, we've been lucky. We've been able to show that rodents have cognitive processes that were previously thought to be beyond them. And this includes the processes that I described in the first slide. We've had various papers kind of like pushing the envelope on what it's known that rodents can do. So that's exciting for us, and we hope in general for the field. We've also, of course, been doing recordings and optogenetics. We're not only interested in the behavior, we also want to understand how the brain implements these processes. But there's been a really big difference in the number of behavioral sessions that we can do per day and the number of electrophysiological optogenetic sessions that we obtain per day. 2 orders of magnitude difference. And at the root of that has been that we need an individual to watch the animals when they're doing the electrophysiology. This is often because when we record from the brains of rats trained on some tests, for example, the rats like to chew on the cables that transmit the signals from their brain to the amplifiers. And, it's very difficult to set it up so that they don't do something that you don't want or chew on something important. So it ends up being important to just kind of watch them all the time. And that's what has limited this, limited us to these numbers. And it's been kind of painful, actually, to have so much of this compared to relatively so much less of this. So to address that, we've been working with some other labs and using some systems that they've developed, in particular from Alla Karpova's lab at Janelia. They've worked on wireless systems, both for recordings and together with a company called Spike Gadgets, which is run by somebody that came from the Karpova lab, did a postdoc in the Karpova lab, also wireless optogenetics. And that has been very important for us. And the reason it's very important is that with these wireless systems, we no longer need those specialized rigs for the electrophysiology or optogenetics. And so that means that we can deploy this across our entire behavior room. Now, of course, once you do that, if you have 30 copies of something, the odds of something going wrong in one of them, once you have 30, now becomes quite high overall. So we need a lot of systems to make sure that the system operates smoothly, and we're working on those. But it is really looking quite good. And so that means that we're looking forward to having many, many more sessions that give us information about the brain. We're now starting to do this with wireless neuropixels, the high-density silicon probes. And so we think that that is going to give us a lot more information about the inside of the brain a lot faster. So we're very excited about that. So the lab works on all four of the processes that I described in the first slide. I'm not going to talk about all four today. They all end up relating a little bit to each other. They all end up using the similar brain structures, an overlapping set of brain structures. Today, I'm going to focus on one story out of executive and cognitive control. And this is being led by a postdoc in the lab, Marina Pagan, who's absolutely fantastic. I hope you'll enjoy his work. And we've been doing this in collaboration with David Susilo and Valeria Mante. So this refers to what I described initially as the ability to route information through your brain and to control how that routing happens. So for example, as you're listening to this, you could either be listening to the speaker saying something, or perhaps much more interestingly and importantly, you might be thinking about some spectacular breakfast that you're deciding to make for yourself tomorrow. And you can choose between these two, right? You can route the contents of either into your awareness. How do you do that? Let me give you a different example. Controlling awareness is difficult, and of course that's something that would be very difficult in animals. Making a controlled experiment for that would not be easy. So let me give you a sensory motor routing example. Imagine that you're at a meeting, a busy meeting, and you hear your name called out. You might want to orient towards the speaker, in which case you might make a decision about how to respond that depends very much on information about the sound location. Or maybe what you want to do is identify the speaker, and you want to make a decision that uses more information about the frequency of the sound, about the frequency content of the sound. You can choose flexibly to make decisions based on location information or frequency information out of auditory stimuli. So that's an example of the kind of thing that we're trying to study. So Marina developed a rat task to study this question. So I'll describe that task to you now. I'm going to take you through one trial of the task. The animals do about 600 trials per session. The sessions last an hour and a half to 2 hours. So those are the typical numbers that we get. And our animals in our behavior boxes, our rats, face a curved wall that has 3 nose pokes in them. So these are little ports with conical openings that the animal can put its nose into, and if it puts its nose into there, it breaks an infrared beam, and so we can detect that its nose in there. And we also have lights in them. And the first thing that happens is that a light turns on in the center poke. And that's the signal to the trained animal to put its nose into that center port and keep it there until that center light turns off. Once the animal has its nose in the center port, we play 2 streams of randomly timed clicks. From a speaker on the left and from a speaker on the right, both streams going at once. Click, 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 click. And the thing is, each of the clicks has 2 features. Each click can either come from a left speaker or a right speaker. And in addition, each click can either be high pitch or low pitch. I don't know if you could hear that over the microphone, but imagine, beep or boop. Okay, so the animals hear that until we turn the center light off. And once we turn the center light off, the animal has to withdraw. And poke its nose either into the right or to the left. And if it chooses the correct side, it gets a water drop as a reward. If it chooses the incorrect side, it gets a white noise, and a timeout, and then the next trial starts. So what defines what the correct side is? Well, there are two different blocks of cued trials. In one block of trials that we call the location trials, what the animal has to do is decide which of the two sides played the total greater number of clicks, ignoring the frequency information. Were there more clicks on the right than on the left? If so, turn to the right. In another block of trials, and these two blocks alternate within sessions, what the animal has to do is whether there were more high-frequency clicks, total high-frequency, than low-frequency clicks, ignoring the location information, regardless of where the clicks came from. Were there more high than low clicks? And if there were more high than low clicks, then turn to the right. So this example of the clicks that you see right here, if the trial is in the location block, that would mean go to the right. That's the correct thing to do. But if these clicks were played in the frequency block, that would mean go to the left because there were more low frequency clicks than high frequency clicks. And the animal has to alternate between these two. As I said, we do them in blocks. In addition, we cue each of the trials at the very beginning with a context cue that tells the animal whether this is going to be a location type trial or a frequency type trial. In this talk, we won't be studying the switching. We'll be studying what happens once the animal switches. They take about 2 or 3 trials to fully switch. So they switch very fast between performing this task and performing this task, but they can perform both and flexibly switch, just as we humans controlling the routing of information. In this block, it's the location information that drives decisions. In this block, it's the frequency information that drives decisions. Now, we can control how easy or hard it is to tell whether there were more high clicks or low clicks by controlling the rate of clicks of one type or the other. As randomly timed clicks, they're Poisson processes, so they're as random as they can be. And for example, if we have 39 low clicks per second on one side, 39 low clicks per second on either side, versus 1 high click per second, then it's going to be very easy to say that this is a predominantly low clicks trial. Whereas, for example, if we have 21 low clicks per second and 19 high clicks per second, that's going to be a difficult trial. And we can titrate this all the way from the most easy, go low, or the most easy, go hard, and everything in between. And independently, we can also do the same titration for the location information. So we have a plane of easy to hard trials. And across different trials, we sample from the entirety of this plane, so to cover the entire plane and help us assess the behavior of the animal. So here's an example of the behavior. I think this is 10 rats. By now, Marino has trained about 60 rats. And let me show you the thin lines are individual rats, the thick lines is the mean across rats. And what I want you to see is that when the animal is in the location block of trials, what we call the location context, the location evidence, the rate of clicks in terms of location, has a big effect on the animal's choices. Whereas the frequency evidence has an effect, but it's a much smaller effect. Contrariwise, in the frequency context, the location evidence has a small effect on the animal behavior, but the frequency evidence is the one that really more strongly drives the behavior. In other words, the animals are performing the task quite well. They really switch between basing their behavior on frequency versus basing their behavior on location quite well. So that means that we can study the process in the rats. So how does the irrelevant sensory information not drive the decisions? How is it that the decision is only driven by the relevant information? Well, one model that's out there in the literature, perhaps the simplest and most immediate model that one would think of, is what if the sensory information that is irrelevant simply never reaches decision-making areas of the brain? What if it's quenched or gated out? So for example, when the animal is in the frequency context, location sensory information is simply blocked from reaching decision-making areas, and of course the decision is going to be driven by frequency. Whereas in the location context, it's the frequency information that is quenched or gated out, and the decision will be only driven by location. That's certainly a possibility, and there's reason to think that that might be the case. In order to address that question, Valeria Mante and David Cicillo, our collaborators, developed a task for monkeys. And I should have said, I forgot to say at the beginning, the task that Marino developed was very much inspired by this monkey task. It's an extension of their work that we're doing. So they developed a task that has a similar flavor to ours. As I said, ours is inspired by theirs. they also had two features of stimuli and they had two contexts. In their case, the two features were motion or color of a field of random dots, and the monkeys were asked to make decisions either about the motion of the dots or about the color of the dots. They trained the monkeys to do the task, 2 monkeys, and then they recorded from the frontal eye fields of the monkeys. And they recorded many neurons, and what they found was that the neurons had highly heterogeneous responses, very, very different responses for each neuron. So in order to analyze these data, they looked at population responses, meaning that they were looking at responses in neural space. So imagine a space in which each dimension is the firing rate or activity of one neuron. So if you had 3 neurons, that would be a 3-dimensional space. If you recorded from 100 neurons, this would be a 100-dimensional space. And a point in this space represents the firing rate of all of the recorded neurons at a given point in time. And so trials become trajectories through space as time evolves. There's a trajectory through the space that is evolving. The space is very high dimensional. If you recorded from n neurons, it'll have n dimensions. So what Mante and Sostillo, working together with Krishna Shenoy and Bill Newsom at the time, what they did was project these data, these trajectories in space, project them down onto special axes so that they could analyze the data more clearly. And the axes that they chose were either axes, directions in space, that best predicted the choice, another axis that best predicted the motion in the stimulus, and another axis that best predicted the color in the stimulus. And when you see systematic variation along these axes, what that is telling you is that there is information about that feature in the neural data. So for example, when you see systematic variation along the choice axis, that's telling you that there's information in the neural activity in the FEF about the animal's choice. There's a lot of information about the animal's choice. You can predict it quite well. And here, when you look along the motion axis, you can see that in terms of the strength of the motion stimulus, there's also systematic variation along this axis, telling you that in the motion context, there was a lot of information about motion, which makes in the frontal eye fields, which makes perfect sense. It's the motion context. The animal is going to base its decision in terms of motion. So you would expect that in a frontal region like the FEF, there would be information about the feature that is driving the decision. The surprising thing was that when they looked in the motion context for information about color, there was also a lot of information about color, even though this feature of the stimulus is irrelevant and according to this hypothesis should have been blocked off and should not have reached FEF. Similarly, in the color context, even though the information driving the decision is color itself, there was nevertheless still a lot of information about motion. So what this told them was that this hypothesis, that the irrelevant information was quenched out and never reached these frontal regions, that's just wrong. So inspired both by their task and by their analysis, Marino ran the same analyses on about 3,000 neurons that he recorded from the rat frontal orienting fields from about 5 rats. And of course, in our task, we were using location versus frequency, not motion versus color, but it's the same idea. He ran their same analysis, and what I hope you'll see is that qualitatively speaking, this looks really remarkably similar to the monkey data. So we reached the same conclusions. In the rats, it is also true that quenching does not explain how the task is being performed. So not quenching, neither in the rats nor in the monkeys, not at all. Okay, so it's not quenching. So what is it? Well, in the task that I described to you, I told you that in either of the blocks, what the animal has to do is base its behavior on total numbers of clicks. Total number of right versus left in the location block, or total number of high versus low in the frequency block. Since it's the total numbers that drive the behavior, that means that as the clicks are being presented, and the click duration is about 1.3 seconds, as the clicks are being presented, the animal has to add up the total number of clicks. It has to be adding up these totals in order to have the totals ready to be able to make a decision. And the general idea about how this happens is that in this neural space, there's going to be some axis, some direction in that space that we're going to call rho, that acts as a line attractor. In other words, that activity, neural activity in that space is stable along it, such that external inputs like the clicks can move you along this line attractor, and one end of the line attractor will represent the decision to go right, whereas the other end will represent the decision to go left. So that is, the accumulated evidence, as it is accumulating, lies along this line. In other words, in the location context, as the clicks are being played, they'll push the neural activity, they'll push the system, the state of the system, back and forth along this line, with right clicks pushing it to the right for each right click, and left click is pushing it a little bit to the left for each left click. In the frequency context, it's going to be the high clicks that push it to the right and the low clicks that push it to the left. In other words, the very same set of clicks can push it in different ways and different directions according to the context that the animal is in. And the mystery is, how does that happen? That's the key question that we're trying to answer. One possibility that you might think of is, oh, maybe the orientation of this line attractor is different in the two contexts. And that difference is the basis for things being pushed around in different ways in the two contexts. So it turns out that they looked for evidence of this in the monkey data, and they found none. In fact, they found that the two line attractors were remarkably close to parallel to each other, and we found exactly the same in our rat data. So it's not a difference in the orientation of the line attractors. Okay, so what could it be? So let's think about this a little more. And I'm going to show you something that is in their 2013 paper, and then we'll start on new things, on new developments. So let's think about this line attractor in neural space, and let's linearize the dynamics around it. Okay, so of course it's a nonlinear network. It's nonlinear dynamics that we're studying, but at any fixed point, and this is a line of fixed points, is a continuous line of fixed points, we can linearize dynamics around it and study that. So let's say that in the absence of clicks and the silences between the clicks, this represents the J as a matrix, and this represents the linear dynamics. Each element of this vector is the activity, the firing rate of one neuron. Now, since this is a line attractor, it means that this is a continuum of fixed points. A fixed point has the time derivative equal to 0. So that means that all along this line, this is going to be equal to 0. That tells us that this vector rho, the line attractor, is an eigenvector of the matrix J with eigenvalue 0. In addition, J is going to have other eigenvectors with their own eigenvalues. But we know that this is a line attractor. The idea is that the activity is stable along this line. And knowing that it's a line attractor means that all the other eigenvalues are negative. Now, let's consider an input pulse that we're going to call I. And the input pulse perturbs you in this space along some direction. And the direction in which the input pulse perturbs you is what I'm illustrating here as I. Let's write this vector I in terms of a linear combination of the eigenvectors. And once we write it like that, we can take this and rewrite it in this matrix form, where this matrix V has as its columns each of these eigenvectors. And this vector E is the vector that has as its elements the coordinates in the eigenspace, these numbers, for this vector I. In other words, this tells us how I is related, I in neural space coordinates is related to the same vector, but in the eigenspace coordinates given by E. And once we have that, we can multiply on the left by the inverse of V. And this tells us if we know the coordinates of I in neural space, we can find the coordinates in the eigenspace, in the coordinates of the eigenvectors. Okay. So that tells us that this top row of V inverse plays a special role, because this top row dotted with input I, that is going to tell us this zeros coordinate, the coordinate that corresponds to position along the eigenvector. Now, for any of the eigencoordinates, the nice thing about the eigenspace, of course, is that they all evolve independently of each other. Fundamental linear algebra. So for any of the eigencoordinates, their evolution according to time is going to be equal to what their value was at t equals 0 multiplied by this exponential dominated by its eigenvalue. Okay. This holds also for this coordinate, E0, the one along the eigenvector. So that tells us that E0 as a function of time is going to be this dot product, this vector S, that we call the selection vector. It's going to be the dot product of S with the input I multiplied by E to the 0t. Since this is 0, this dies away. And so this tells us that this dot product is a constant throughout the evolution of the dynamics. Now there's all the other eigencoordinates, of course, but they all have negative eigenvalues. So over time, they all die away. So that means that after these transients have died away, what tells you how much this input I moved you along the line attractor is this dot product, S dot I. So if you know this, that's all you need to know where you're going to end up. Now note that Note that there's also those other transients. So your initial position along the line attractor isn't necessarily equal to this. That's what you get only after the transients have died away. Okay, so that tells us that the important thing to look at is this dot product. That's what tells us how much we moved along the line attractor. So if we have two contexts, how much we move, an input pulse moves us along, for example, an input pulse of location information, how much it moves us along the line attractor in context 1 is given by this. How much it moves us along the line attractor in context 2 is given by this. So the difference in the two contexts is necessarily given by the difference between these two things. Right? So if we want the pulse to affect the choice differently in the two contexts, this should be non-zero. And this applies for any system in which decision evidence is accumulated on line attractors. One thing that's appealing about this analysis is its generality. And now I'm going beyond what was in the 2013 paper. Now I'm starting to tell you about the work that we're doing now. So we realized that you could take this and you could rewrite this in the following way. Take this first selection vector, the selection vector in context 1, and just rewrite it as the average of the selection vector in the two contexts plus the difference across the two contexts divided by 2. And similarly, S2 can be written as the average minus the difference divided by 2. And once you've written it this way, you just expand this, and what you get is that the difference in context 1 versus context 2, the difference in how evidence is accumulated, is equal to this. And this, in a way, is very simple and interpretable and feels very natural. This is telling you that if you want to have a difference of how a pulse affects you in the two contexts, then you could either have a difference in how the input reaches the decision-making areas, how that sensory information reaches the decision-making areas, or you can have a difference in the dynamics in the decision-making areas across those two contexts for a given input pulse. Right? And the quenching hypothesis was a version of this. Right? In the quenching hypothesis, the difference across the two contexts was that the magnitude of I in one context was 0, it was quenched out, that was a sensory input, and in the other input, it was not quenched out. So we've ruled that hypothesis out, but there's many changes in this vector I across the two inputs that don't depend on the magnitude being different. Now, in addition, if the line attractor direction is the same in the two contexts, in other words, the two line attractors and the two contexts are parallel to each other, which is what we believe, then you can further break this term down into these two terms. You can look at this average selection vector and just ask what is the component of that parallel to the line attractor? We're going to write this as S rho, parallel to the line attractor. And what is the component of S bar that is orthogonal to the line attractor? So this just gets broken down into these two bits. Both of them depend on input modulation. The input is changing across the two contexts, but they'll have different implications for biological variability that I'll tell you about in the next few slides. We call this one direct input vector modulation, and this one indirect input vector modulation, and the reason for the names will become clear in a second. And this one we call selection vector modulation. Selection vector modulation because it's a selection vector that changes across the two contexts. These two is the input vector that changes across the two contexts. Now note that to accumulate evidence differently in the two contexts, this has to be non-zero. And the description I've just given you, if these two conditions apply, if decision evidence is being accumulated on line attractors and the two line attractors are parallel to each other, then every linearized view, and of course linearizing around the fixed points makes sense, must use this. This must be true for any network, and therefore any linearized network must use some combination of these three possibilities. Right? It's the sum of these that tells you what the total effect is going to be. And if what we're interested in is the relative contribution of them, then we could say, oh, this sum is going to be equal to 1. We normalize that. And if that sum is going to be equal to 1, then every network must lie somewhere along this triangle, where your position along the triangle tells you about how much you're using this type of approach versus this type of approach versus this type of approach. For example, if you're in this corner, that means it's all this and none of the other two. So any network can be placed along this triangle, and it'll tell us a lot about how the network is being able to perform the task, how it's able to integrate information differently in the two contexts. So let me illustrate the different types of approaches. Let me illustrate different positions on the triangle. Let me first illustrate this corner, what we call direct input modulation. So imagine that in one context, this is a selection vector, the line attractor, this is the input vector. And so an input would move you here, and then if you follow the gray lines, you follow the dynamics, say this is an input of location information, in the location context, it might end up moving you along the line attractor quite a lot. But in the frequency context, let's say that the input vector moved this vector relative to that one, has moved parallel to the line attractor. That's how we defined this corner. And because of that, the relaxation after the input pulse is different. And now you'll see that this input pulse brings you back to the origin. You don't end up moving from where you were, whereas this one moved you further. If we look at the projection along the line attractor, so along this choice axis as a function of time, you can see that in one context you end up moving a lot, in the other context you end up coming back to the origin. And so that's what gives you the difference across the two contexts and how information is integrated. I also want you to notice that the difference in the two contexts is immediate along this axis. As soon as the input pulse is presented, you can see it because the input pulse has different projections onto the line attractor, onto rho, and that was by design, right? We're looking at this extreme case in which the change in the input vector is parallel to rho. So let's look at the other two cases. Now let's look at this selection vector modulation. In this case, what happened, the difference between the two contexts, was not that the input vector changed, it's exactly the same, but that the selection vector changed. The direction of the selection vector has changed. And because of that, the flow after the input pulse is different in the two contexts, and that's what gives you the different effect in the two contexts. But now notice that at the beginning, the two contexts produce exactly the same effect because, of course, it's the same input pulse in the two contexts, so its projection onto rho has to be the same. It's only afterwards that the difference develops. And finally, this corner, in this one, the selection vector is the same across the two contexts. And now the difference in the input vector along for the two contexts is not parallel to the line attractor, but is orthogonal to the line attractor. This input vector has moved vertically down to this. Once again, this means that because the projection onto the line attractor is the same in the two contexts, the immediate response to the pulse is the same in the two contexts, and the difference across the two contexts builds up only gradually over time. So that tells us that this axis, this vertical axis, this one really tells us about the fast versus slow response of the system. Up at this corner, context effects are immediately felt, whereas down here, they take time to develop. In addition, this axis over here is telling us about how much is the process dominated by modulation of the recurrent dynamics, modulation of the selection vector S, versus modulation of the input. And this, of course, is going to be very important because this can correspond directly to an anatomical understanding of how this works. Approaches that lie at this end could function by changing the feed-forward input, the feed-forward connections from sensory to decision-making regions, whereas this approach lies in modulating things within the decision-making regions. So this has implications for the anatomy. This has implications for the speed of the process. And as I'll tell you, the second axis, fast versus slow, matters for understanding some of the biological variability that we see. Going back to what Monte and Cecilia did in 2013, they recorded all these data from the FEF. They decided that quenching was not the answer. They also decided, they didn't just decide, they showed that change in the line attractor was also not the difference between the two contexts. So to try and get a better understanding, they used backprop to train large recurrent neural nets and analyze how those recurrent neural nets, once they were trained and were performing the task, how they did it. And what they found was that the recurrent neural nets predominantly use selection vector modulation in order to perform the task. It changed in the dynamics across the two contexts. They then took those recurrent neural networks and analyzed them in the same population space approach that they had used with their data. And what they found was that the recurrent neural networks and the monkey data were qualitatively quite similar to each other. So that led to the natural suggestion that selection vector modulation was a leading candidate for how context-dependent decision-making takes place. Nowadays, now we understand about the geometry of this triangle, so we can take those old 2013 recurrent neural networks and place them on our triangle because we can measure the selection vector and we know what the input vector is. for any of the networks, and so we can measure the changes across them. And it turns out that if you do a heat map, you train many hundreds of these networks, and you place each of them on this triangle, and you ask where they go. This is a heat map of where they land. And indeed, they're not all exactly at this corner, at the selection vector modulation corner, but that's where they tend to go. If you asked which corner are they closest to, it's clearly the selection vector modulation corner. This is the predominant term, exactly as they said in 2013. However, armed with this understanding of the triangle, we're no longer limited to what backprop tells us. We don't have to only study the networks the backprop makes. Armed with Marino's smarts that led us to understand how to do this, we can now build a network. We can engineer a network, a large recurrent neural network that successfully performs a task. We can engineer it to be anywhere on this triangle. So the heat map for networks, large networks that perform the task in our hands now looks like this. We can make a network anywhere in the triangle. Okay, so armed with this new knowledge, now we can make networks anywhere on the triangle and ask, if we run this analysis, how much do they look like the data? And get a much better sense, instead of the narrow backprop trained view, get a much better sense over the entire triangle of how things work. And remember, once you've said quenching is not the answer and line attractors are parallel in the two contexts, this triangle represents the entire universe of possibilities. So we're really interested in figuring out which parts of the triangle apply and which don't. So here's a recurrent neural network that lies at this corner of the triangle. And here's what it looks like in terms of this analysis. And here's a neural network at this corner of the triangle. And here's a neural network at this corner of the triangle. And what I'm hoping you'll see is that all three of them qualitatively look just the same like these two as these two panels. There's not one of them that you would say is closer to these panels than the other. I'm only showing you the location information, but it's something very similar happens to the other one. In fact, across the entire triangle, the recurrent neural networks look very, very similar to the data. So what that tells us is that in terms of this subset of possibilities, we don't really know where the biology lies yet. So the state of play at this point then is that the quenching model was ruled out by Mante and Susilo in 2013. We found the same in our rat data. A change in the line attractor across the two contexts was ruled out by Mante and Susilo in 2013. We found the same in our rat data. But pretty much everything else that remains is still on the table. Now, I also told you that, and I'd like to point out that this space is a very important space. I already told you this axis has very important implications for dynamics, and that this axis is going to have implications for biological variability. So figuring out where the biology lies in this triangle, we think is very important if we want to understand how this works. I told you that our task is a pulse-based task. And I also told you when I was developing this analysis of understanding the geometry of this triangle, that understanding how pulses affect dynamics around the line attractor was very useful in understanding it. So because our task is a pulse-based task, we can leverage those pulses to analyze the dynamics of the neurons as they are being recorded. And that's going to give us a lot of leverage in terms of placing animals within this triangle. As I'll tell you, we think that we've gotten a good handle on where each animal lies along this axis. This axis is harder and is something that we're still working on. So in order to pursue this approach, what we need to do is to estimate the response to single pulses. So let me tell you how we do that. Let's take a particular train of pulses. And these could appear, we can look at what the location evidence in these pulses is in the location context, or we can look at the location evidence of those pulses in the frequency context. Separately, we could ask, what's the frequency evidence that these pulses carry? And we can ask about that in the location context or in the frequency context. And we're going to treat these four cases separately. And then each neuron's firing rate, we're going to model it as if each of these four types of situations has a kernel that describes the firing rate. In other words, we're going to multiply every time that a pulse like this happens, we're going to add this function of firing rate as a function of time to the modeled firing rate. And we're going to have a kernel for location evidence in the location context, a kernel for location evidence in the frequency context, one for frequency evidence in the frequency context, and a different one for frequency evidence in the location context. So we have all of these kernels for all of the different types of pulses. In addition, we have an overall firing rate as a function of time that we can also adjust, an estimate of context effect as a function of time, and an estimate of the ultimate choice of the animal as a function of time. And all these kernels, we're going to try to search for a fixed set of kernels that best match the firing rate of the neuron across all the trials. So the predicted firing rate is going to be the sum of all of these. So here's an example neuron. I'm showing you the predicted firing rates for two different choices in black and red. And here's the actual neurons data. And what I hope you can see is that for this example neuron, these match quite closely. So what that is telling us is that this is a pretty expressive model, so it can describe the data pretty well. And these kernels, these are our estimates of the single pulse responses. So that's how we get the estimates of the single pulse responses for individual neurons. We can do that for each neuron, and then we can plot these in the neural space. We can plot the kernel as a function of time in neural space, and we can project that onto the choice axis, just as I was doing with the cartoons that I showed you before. So in the triangle that we had those cartoons that I showed you in the movie, and that for the direct input vector modulation, there's an immediate change in the pulse kernel according to context. So this is pulse for a particular type of information. For example, this might be location information in the two contexts, whereas in these two approaches, the change is much more gradual. So let's ask what happens in the data. So in the data itself, here's what we estimated for the pulse responses and the projection onto the choice axis as a function of time for one rat for location information. And you can see that this looks qualitatively quite similar to these cartoons, right? The effect of the pulse initially is the same in the two contexts, but then gradually differs according to whether location is relevant or irrelevant. So that would put this kind of somewhere along this line, right? It's in the slow edge of things. If we look over here for another rat, analyzing the responses to frequency information, this one, there was an immediate difference according to whether frequency was relevant or irrelevant in the frequency kernels. So this looks much more like this cartoon, right? So that would put this over in this corner. And in fact, as we looked across animals and across frequency and location, we found pulse response kernels for relevant and irrelevant information that lay everywhere along this axis. In other words, a lot of variability across the animals, even within an animal. So for example, an animal could be like this for location, but like this for frequency. So what does that tell us? Well, the first thing it might suggest is, well, you just have a noisy measure. You're seeing a lot of variability because things are noisy. But actually, we know that it's not noise, because if we take the data and cut it in half and do the analysis for each half of the data, we get very similar responses for the two halves. So it's not noise. So what else could it be? Well, we were struck by we saw variability in these neural responses, and we're also struck by variability in some of the behavioral analyses that we had done. So here I'm showing you data from one particular rat, and I'm showing you the psychometric functions. This is for location, when location is relevant, affecting the choices a lot, and when it's irrelevant, affecting choices much less. Similarly for frequency. And I'm also showing you what we call the context-dependent psychophysical kernel or behavioral kernel as a function of time. What we do is at any point during the stimulus, we take the difference in the clicks for right versus left and use that in a logistic regression model where different time points are different elements, different regressors for the logistic regression to predict the choice of the animal. When we do that, that gives us a weight for how important are clicks at any point in time in affecting the ultimate choice of the animal. And when we compare across the two contexts, that's what I'm showing you here. I'm showing you the difference in the context-dependent kernels. And what you can see is that for this animal, the difference for location-relevant and irrelevant information is not very large, whereas it's much larger for frequency. And this actually matches what you see in the psychometric curves, in that the performance for this animal in location is not that great. It is quite affected by location, even when location is irrelevant, whereas it does much better at frequency. The effective frequency when it's relevant is much, much less than when frequency is relevant. Right? And that matches the differences that you see here. Now that's one rat. Let's look across 20 rats. And if you look across 20 rats, it's striking how different the context dependent is. There are some rats, say like this one, in which there's a strong context-dependent effect in how the animal is affected by clicks from the beginning of the stimulus all the way to the end. There are other animals, for example, like this one, where there's an important difference at the beginning, but by the end there's almost no difference. And it turns out that if we thought about it, this makes sense in terms of the fast versus slow context effects that we are finding in the neural responses. So imagine that the animal is making a decision based on these clicks, and at the end of the clicks, it's asked to report its decision. Well, if it's using this fast mechanism, if it has these fast neural effects, these final clicks are going to be treated differently in terms of which context it's in by the decision time, which is very soon, and they can be treated differently because the context effects kick in very fast. Whereas if it's using this more slow context effect kind of approach or mechanism, then the context dependence of the final clicks won't have had time to kick in before the animal has to make its decision. So if we look, for example, at location evidence across the two contexts when location is relevant versus irrelevant, and ask how much do these final clicks, how much are they correlated with the decision? We do that logistic regression analysis, and we compare across the relevant versus irrelevant conditions. This is a behavioral measure of how much those final clicks affected choice. And we can take it as a behavioral measure of how fast the context effects kick in. And remarkably, when we correlated that behavioral measure with the neural measure that we had measured, and actually we measured the two in different sessions. So these were purely behavioral sessions, these were recording sessions, but in the same rats, we find this very, very, very high correlation. The neural and behavioral measures are highly correlated. So that tells us that that fast versus slow axis explains a lot of the variability that we see in the behavior and has strong biological implications in terms of affecting how that behavior comes about. And so I've told you that we think that we've got a handle on placing each animal on this axis. How about the other axis, this axis? There, things are more difficult. I'm not going to go in detail into what we're doing. I'm just going to tell you that what makes this difficult is that it requires estimating the flow and the dynamics. This one, really, we could get at just through this fast versus slow, which we could see in the choice axis. And the choice axis corresponds to this a component of the selection aligned with the line attractor, and it's relatively easy to estimate, whereas these other components of the selection vector, these general dynamics, are much harder to estimate. So to try to do that, we're using simultaneous recordings from neuropixels to, on a single trial basis, estimate what the flow is and reduce the dimensionality simultaneously so that we can get a decent estimate of that flow. And what we're hoping to do is, through these estimates, actually get direct estimates of what the flow is, and therefore direct estimates of the selection vector and possible changes across the two contexts. So that's what we're trying to do. So what have I told you today? First, that we developed a method to efficiently train rodents in dynamic context-dependent selection and using different features from a sensory stream. We have enough trained individuals that we could assess individual variability. Getting this task to work in rodents now opens the door to the use of molecular tools that are more easily available in rodents than non-human primates. We also developed a new geometric understanding of the space of networks for context-dependent decision-making. This should say depth, not deep, sorry. And that geometric understanding, it was very important to allow us to look at large networks that went beyond the ones that were trained by backprop. Backprop gives you a very narrow view of possible networks. There's a bigger world out there that's compatible with the biology, and that can be important to consider. Here we were lucky that Marino figured out this triangle, and that allowed us to then explore the entire triangle. Individuals can use different algorithms to achieve the same computation. You saw that in that variability across the fast versus slow axis. And that can be an important issue in cognition, where the algorithms can be purely internal. Different animals could use different internal algorithms to perform the same task. Often, only the findings that are consistent across animals are reported, but that can obscure meaningful real biology. And so that's a real issue. So we need ways to measure and characterize each individual's computations. And I would argue that using these kind of pulse-based tasks That's an idea that comes from engineering, using impulse responses for system identification, and we're finding that it's an idea that is very useful in biology too, not just engineering. So finally, where are we going with this? Well, I told you at the beginning about our efforts to do this high-throughput electrophysiology and optogenetics. So we're hoping to record not only from the FOF, but also from sensory and many other brain regions, and hopefully get to understand the brain-wide networks that support flexible feature selection in this task. Finally, I'll end by once again highlighting Marino and our awesome collaborators. They've been fantastic to work with, David Sosillo and Baleramante. And many other people have contributed, people in the lab, both former people and current people in the lab that contributed in various ways. So I wanted to at least put their picture up here at the end. And with that, I'll stop, and I guess we have a few minutes for questions.
Okay, thank you so much, Carlos, for the interesting talk. So we have, as Carlos said, a few minutes for question. Unfortunately, we don't have the opportunity to have the panel with Carlos, which would have
Yeah, I'm sorry about that.
Yeah, more extensive time. If there's any questions, please raise your hand or type it in, so he should be able to see text. So Dr. Sanayi, please.
Hello, Carlos. Thank you for a great talk. Question. In your previous works, you also run the same task on humans. I mean, on your science paper, for example, 2013. Have you collected data from humans on this kind of contextual task?
No, not yet. Great question and interesting to do. We haven't done that yet. And, you know, we imagine that we would see the same kind of variability in the psychophysical kernels, but we have not yet done that, and so we don't know that we would.
Thank you.
Okay, hang on. I see some questions coming in. In the line attractor dynamics, the choice axis is regarded as an invariant direction for V. Why is the choice axis an eigendirection for the main system before projection? So to be clear, when we train recurrent neural networks to do the task, the line attractor is not always lying along the choice axis. They can be a little different from each other. They can be actually quite different from each other. The only thing that matters is that their dot product not be zero, right? So there's a component of the line attractor along the choice axis. And then you can project onto the choice axis and read out the choice. In those same recurrent neural networks, when we do the pulse-based analysis that I described to extract the pulse kernels, we also extract a choice kernel. And we found that that choice kernel, the projection of that in neural space, that is a very good readout of the line attractor, of the true line attractor in the recurrent neural networks. So we're taking that as a good readout of the line attractor in the data as well. So in short, they don't have to be aligned. They just have to have a dot product that is not zero. And we have a good estimate, we think, of the true direction of the line attractor. If that doesn't answer your question, please ask again. Another question is, the manipulation for frequency has two values or change parametrically. It's just two values, either high, each click is either high or low, and the parametric change is really in the rate of clicks. So in the same that each click either comes from the left or from the right, and what we titrated was the rates, the relative rates, and that can be titrated smoothly. We did the same for frequency. That means that we only have 4 types of clicks, and it kind of it simplifies the analysis a little bit, but it still gave us something that was titratable. So that was the approach that we took.
So I have a quick question too. So my crude understanding of Valerio Monte's paper was that what changes between the two conditions is the selection vector. Is that right? But so basically for the same input, for the same eye, it can be projected. The different information can be extracted basically to fall on the line attractor. But exactly. then you have delta I. So that means that you assume that there are different inputs. What does that mean?
Okay. So in their paper, what they found was that most things fell in this corner, right? So it meant falling in this corner means the input is the same,
right,
in the two contexts, but there's a change in the selection vector. But other possibilities are this and this. So I think that's what you're asking about. So
Yeah.
that could be implemented either by a top-down signal that, say, affects the pathways from sensory information to FEF. That could affect delta I. Or it can even happen within FEF itself. So one of the things that happens...
somehow you're imagining that some other region extracts two different information, let's say color versus motion, and then give it to FEF. So that's how delta I is created. So you're just
there are two ways to create delta I.
Okay.
There are two ways to create delta I. Let me see.
Okay.
So forget about the actually, this is a model of delta I, right? So in this approach, delta I is, in one context, it's 0, and in the other, it's non-zero. But in addition, you can create a change in I in the FEF itself in the following way. I represents the linearized input. So at each of the line attractors, we're linearizing along the line attractor. And the two line attractors, although they're parallel to each other, they're not exactly in the same, they're not overlapping, they're not in the same space. They're parallel, but they're separate. That's true both in the electrophysiological data and in the recurrent neural networks. That means that when you linearize along them, although the line attractor is the same, the linear dynamics around them might be different, and that means that I can be different also. So in fact, when we engineer networks to lie anywhere on that triangle, what we do is we first train a backdrop network like this, so the input vector to the network is constant, and the only thing that we're filling with is where the set point is going to be, and that changes the effective, the linearized I and the linearized delta I, and that's how we can place ourselves anywhere.
That means the matrix J, which was the, I don't know, Jacobian or the whatever, so that is, you think it's different for the two contexts. So somehow the system is able to use two different J's for the, I mean,
and in fact, when you're saying that you're in this corner, when you're saying that there was a change in the selection vector, that's where it came from, right? Because the selection vector is a function of J, so for S to be different in the two contexts, J must be different in the two contexts.
But in both contexts, J should have the same null space, the same eigenvector with the eigenvalue 0, right? So the eigenvector with eigenvalue 0 should be the same. That means that is the argument for the parallelness of the two.
Yes, and that doesn't have to be true. That just turned out to be true in the data and in the recurrent neural nets.
Right.
So actually, this triangle view, we have a generalization that takes into account what if the line attractor direction is changing, and then instead of living in this two-dimensional space, you would live in a three-dimensional space. Right. But once again, you can describe all the possible networks.
Very nice.
Thank you. Okay, somebody's asking whether there's time for more questions.
Is there time for? So that's a question for you. Is there time for more questions?
I guess I can take one more question, and then unfortunately, I'm really sorry that I have to go and can't stay for the panel and more questions. I'd love to, but I have to go. Okay, last question, please.
Hi, thank you. So do you think so there is a kind of sequence or kind of a way that the rats are trained, so they can be trained in different ways? For example, when during the training time, they can first get trained about frequencies, and then the other one, or they can be just trained the main task. So I'm saying different strategies can be used to train the rats. So I was first thinking, are there strategies to make the training phase faster or not? But then the question developed into, does that can that affect where the rats end up in that triangle? Is it clear what I mean?
Yeah. Excellent question. And what I'm understanding is two parts. One, how you train the rats, does that affect where the rat ends up on this triangle? And part two was, are there tricks for training the rats faster or slower? And the first answer is that we've looked, so, the task was developed over time, so we actually train rats in slightly different ways over time, and we've looked to see whether there's any systematic difference in where they lie, and we haven't been able to find anything about how we train them that predicted where they lay. In addition, individual animals can say treat frequency over here, but treat location over here, individual animals. Actually, I shouldn't point to the corners. All we can identify so far is along this axis. So that was the same individual animal, right? So what we think is happening is that anywhere within the triangle performs the task. So really that's something that the task isn't determining. And so that leaves the system, the neural networks, free to land anywhere here that successfully performs the task. So in short, Great question, and certainly a possibility. So far, we've found no evidence that the training affects where the animal falls. Finally, on faster versus slower, yes, we've been working on trying to get the thing to work faster. Right now, it takes about 2 months per animal to train the rat. Okay, and I see there are more questions which I'm too tempted to answer to vanish. I should go, but I really want to answer the questions. Have we considered the dimensionality of representations in responding to location versus frequency stimuli? Great question. Not hugely, partly because we know that there's only a few dimensions that matter, right? So really the only dimensions that matter for any particular input type are the line attractor direction, the selection vector direction, and the input vector in the two contexts. So for any particular type of input, there's only 4 dimensions that really matter. So far we've focused on trying to identify those and looking within that space. But the broader question of how does dimensionality play into this task, that's a great question, and it's just not one that we've really explored. So the answer is we haven't really considered it, but we should. And finally, the last question, and after this one I really will go, can the animals switch per trial? Not quite. It takes them 2 or 3 trials to fully switch. So from the very first trial of a block switch, we see a change in the behavior. So that tells us that the cues are informative to the animal. It's not just the fact that the trials are in blocks. And it only takes them 2 or 3 to switch. So it's very fast, not fully implemented on a single trial. One thing that we haven't yet done is actually place the animals on trials where it randomly switches from one to the other. In other types of tasks, when we've done that, if we train in blocks, they have a few trials to switch, but then once we train them in fully randomized, they're able to do it. We don't yet know whether these animals could do it in this one. I suspect yes, and it would be interesting to see that it could be that fast. All right.
Okay.
Thank you for the great questions, and my apologies for having to go.
Okay, we were excited to see you, Carlos, and best of luck for the research, and hope to be in touch with you. Thank you.
Yes, please, and thank you very much for having me. It was a pleasure.
Okay.
Bye, everybody.
Bye.